Hello everyone and welcome to the first of our Pracs You Can Do at Home series. Today we are going to be looking at how we can extract DNA out of various organic materials. Now when people think about DNA and the sort of work that you do with it in laboratories, they might think that it all might need to be done in highfalutin molecular biology labs. And for the most part it does, because it needs to be safe, it needs to be kept clear of contamination and the like. But the very basics, pulling DNA out of a living thing, can actually be done with materials you can find lying around your house. Let's get started. I've amassed some equipment over here. The first thing we're going to need is something to pull DNA out of, and in this case, as the backdrop suggests, we are going to be taking it out of a strawberry, a living thing that has got DNA inside there. Now we need to have something that we can use to break apart the strawberry and to release the DNA into solution, and the best thing for that is a Ziploc bag. Basically we put the strawberry and our ingredients in there and mash it up as much as we can. The next thing we're going to need, one of the ingredients we're going to have to include in there, is a detergent of some description, and dishwashing liquid works well for that. In a laboratory they tend to use SDS, so it really doesn't matter the quality of the dishwashing liquid that you use. The cheaper the dishwashing liquid, generally the more SDS it's got in there. Any sort of dishwashing liquid will do that, and that's there to break up the membranes of the strawberry cells. We then need to filter most of the chunky bits of strawberry out, and to do that, a Chux cloth. Okay, that is certainly just as good at doing the job as a piece of filter paper. We then need something to extract the DNA out of the water, and we generally do that using alcohol. Now the gold standard for DNA extraction generally uses the alcohol isopropanol, which can be found in isocol rubbing alcohol, but at current state it's very difficult to find isocol rubbing alcohol in the supermarkets, so we're going to settle for another alcohol that's reasonably easy to get at, and that is methylated spirits. We tend to use isopropanol because it's a little bit safer. I mean, they use it as a rubbing alcohol on the skin. Methylated spirits can sometimes have some nasties and contaminants in it, so I just recommend you take some care with using the methylated spirits. Apart from that, just a few little bits of glassware and we should be right to go. So let's get stuck into it. Let's extract DNA from a strawberry. Okay, so the first thing we're going to do is we're going to need to get the strawberry ready. Now the leaves tend to get a little bit in the way of the crushing process, so throw them away. Now for a bag of about the size that we're going to be using, I reckon we can probably get away with just half a strawberry. So all we do for this is open up our bag, place our strawberry in there. The next thing we do is just a small squirt of dishwashing liquid in there, just a little bit. Alright, next up, we need to break apart our strawberry cells and release the DNA that's found inside. Now, the best way to do that is to add a little bit of warm water. So, if I can just tip that in there. You don't want to fill the bag right up. Now, a bit of a trick for this. We need to get as much of that air out as possible. So just be very careful, squeeze as much out as possible. That just makes the squashing action a little bit easier the next time around. So now we have our strawberry in nice warm water there. Now there's actually another step in between adding your detergent and your warm water, your strawberry, and squashing it up, and the next step, which is pouring it into the container to do the extraction on. And that's the addition of a protease enzyme. Protein can interfere with what we're about to do, and so particularly in real molecular biology labs, they quite often use a protease enzyme to get rid of that protein so it doesn't get in the way of what they're going to do with the DNA. Now, it used to be very easy to find a protease enzyme around the kitchen. In the spice section of your supermarket, there was a little bottle of something called meat tenderizer. And it was an enzyme called papain, which comes from pawpaw seeds. And it breaks down protein like any good protease does. And so you would just add a little bit of a pinch to the mixture and mix it all in when you're mushing up the strawberry. Unfortunately, it seems to have disappeared from the spice section of the supermarket. I have seen it in some Asian supermarkets, so you might be able to find some there. But in the long run, it really doesn't matter if you're just doing this from home. It's really just there for completeness if you really want to make this like a real-life DNA extraction. Out of the way. All right. Now, this is the bit where you can rope in a younger sibling or something similar, because this tends to be the bit that they enjoy the most. Now, what we have to do is we have to just start squashing that strawberry up. I may have had too much water in there. Now, the idea of this is to break apart as much of the strawberry as we can. Try not to burst the bag as well, because that would be disastrous. There's 2 points in this experiment where things are likely to go wrong. This is number one, and that is because you tend to open up the bag and send strawberry juice everywhere. That's probably looking pretty good there. All right, now it's time to transfer our strawberry mush into a container that we can do the last part of the extraction on. So, now being at school at the moment, I'm very lucky to have some real life scientific gear here. So I have a fairly large bore test tube there. Now, you are very unlikely to have one of these at home. So, you can do this on anything so long as it is reasonably narrow and as long as it's transparent, because we're going to be able to see the DNA through there. So a narrow glass would work. It just means you've got to use a little bit more of the reagents when you're doing it. But you should be able to find something at home that you can do that in. So I'm going to put that tube in there. I've just cut a small square out of my chucks cloth, and I'm just going to poke it in the top like so, just to make a little bit of a filter. Okay. Now, this next bit, I said before, the place most likely for an accident to occur was when you were squashing up. The second most likely place is this bit, because if we're going to be spilling stuff, it's going to be in this section here. So, all we do for this is we open up one corner of our Ziploc bag and then hold it by the opposite corner, and then you can just by tipping gently, you can pour a little bit of your strawberry in there. Now, I am very experienced with this and I'm still spilling it everywhere, so don't feel bad, just make sure you clean up after yourself. All right. That should be enough. It's really only a few centimetres on the bottom that you're going to need for this one. So like that. So we're now ready for our last part. Now, in our last part, we need to pull the DNA out of solution. So at the moment, the DNA is all dissolved in that strawberry juice you see there. But we need to pull it into something where it's less soluble, and that's where the alcohol comes in. Now, this next bit takes a little bit of skill and practice. I'm going to see if I can get it up a little bit closer to the camera so you can see exactly what I'm doing. All right. For this, we're going to need our alcohol. Now, the alcohol needs to be very, very cold. Okay, so put it in the freezer to chill it right down, and then until you're ready to use it, put it in a little ice bath, just to make sure that it's around about 0 degrees. It needs to be very cold. Now, what we're going to do is we're going to add this alcohol to the strawberry juice, but we're going to do it in such a way that the alcohol forms a layer on top of the strawberry juice. So you should have pink strawberry juice down the bottom, colourless alcohol lying on top of it. So, once again, you can change it into your own little container of methylated spirits. This is just some we had lying around the lab. All right, now, try to find the best orientation for this. Focus. There we go. So the idea of this is to dribble the alcohol down very carefully. It helps if you hold the strawberry juice tube on a slope, but if you dribble it down, eventually it should form a layer that sits on top of the strawberry juice. There we go. All right. Now, I can actually see some DNA coming out in that already. If you look very carefully around the top there, can you see some little bubbles, maybe some stringy stuff there? That is all you need to make a strawberry. Well, plus some proteins and nutrients and things like that. But those bubbles and that stringy stuff, which is not in very good focus at the moment, is the DNA coming out of solution from the pink strawberry juice there and going up into the alcohol on top. Now we can try and pull some of that DNA out as well. Okay, now I'm going to try and pull some of that DNA out. Once again, there's not a lot in there. You can see a little bit around the top there, but we'll see whether we can extract some of it out. Now, the best way to do this is to just get a skewer stick, something similar, and just start twirling that DNA around the top. And you'll actually see it start sticking to the end of the skewer stick. You can sort of scrape it down a little bit into the other bit. Oh, look at that. That's a good bit of DNA there. So, let's see if I can put it on there. A lot of people want to look at it under a microscope. Don't forget, DNA is way too small to be seen under the microscope. The best way that I find to describe it to someone who's never seen it before is kind of like what snot looks like in a swimming pool. I know that's disgusting, but that's basically how it looks to me. Probably says a lot more about me than anyone else, but essentially, and that's sort of dissolved a little bit now, but you could see the big gob that's sort of wrapped around the end. What you've got here, DNA is a long stringy molecule, and those strings are tangling around each other to make this stringy glutinous mass that you see in there. That's how you pull DNA out of a strawberry. So there you have it. That's how you extract DNA from any sort of organic material. Whether you're doing it from a strawberry in your own kitchen, or whether you're doing it from a medical sample in a highfalutin molecular biology lab, the steps are the same. You start off with your sample, you break it apart in the presence of a strong detergent, which takes apart the cell membranes around the outside of the cell, but also around the nucleus, which releases the DNA into the solution. You use a protease enzyme to get rid of any protein which might interfere with the process. You then filter out all the chunks that you don't really need, and then you use alcohol to separate the DNA out. From here on in, you can take the DNA, you can purify it using various other methods, And then you can do a whole bunch of things in the laboratory that involves DNA. You might use it to make a template strand for PCR. You may use it to do sequencing. But the first step in all DNA work always starts with this, extracting it from an organic source. Feel free to experiment with other organic sources of DNA. Different sorts of fruit are really good. Bananas, kiwi fruit, normally have quite a bit of DNA in them as well. You might want to see which one of those fruit this method is best for getting DNA out of. Try to stay away from animals, simply because you can take DNA out of the fruit and they don't mind. Do that to an animal and you're starting to get into more than an ethical grey area there. But certainly the techniques are the same. So hopefully you'll be able to try this at home. Get in touch with me, let me know whether it worked for you, let me know whether you found something that's even better than strawberries to get DNA out of. Thanks very much.